Alright, this video focuses on sexual reproduction in the process of meiosis, gametogenesis, and fertilization. Alright, meiosis is the reduction division process. It's also called gametogenesis. So you can use those terms synonymously. Meiosis is another kind of division. We learned about mitosis already, so now we're talking about meiosis. Remember that somatic cells, which are cells that are in your body, any other cells that are not sex cells, meaning not sperm and egg, they're 2N, which is diploid. That means they have 2 copies of a chromosome. It's diploid. Gametes, which are your egg and your sperm, those are haploid cells, meaning they're just 1N. So there are 23 chromosomes in your gametes, and there are 46 chromosomes in your somatic cells, all the other cells in your body. Sexual reproduction, as you probably know already, is when the egg and the sperm fuse to form a zygote, which is a new individual cell. Each gamete provides 1 copy of all genes for the species. So each gamete, whether it's an egg from the mom or a sperm from the dad, they have 1 copy of all the genes for that species. And they get the other copy from, the other parent. So the zygote has 2N, which is 2 times 23, pairs of all genes. Alright, so in meiosis, there are 2 divisions. In mitosis, when we talked about that earlier, there was only 1 division. So it pretty much goes through the steps twice. So there's 2 divisions. The first division process divides homologous chromosomes. The second division process divides sister chromatids, and they produce gametes with 1 chromosome of each type. So at the end, we're going to have those sex cells, the sperm or the egg, with only 1 chromosome of each type. So the division stages are similar to mitosis, the ones we learned, prophase, metaphase, anaphase, telophase. The division stages are similar to mitosis, so you have meiosis 1 and meiosis 2 for 2 divisions. All right, so basically you have prophase 1 in meiosis 1, and then you have prophase 2 in meiosis 2. So you can kind of read down, all of them are the same: metaphase 1, metaphase 2, anaphase 1, anaphase 2, and so on. Alright, so here's like a diagram for meiosis 1. You have those 2 homologous chromosomes at the very top in the circle right here. You have 2 homologous chromosomes, okay, and there are 46 chromosomes there. The meiosis 1 reduction, after the first step of meiosis, it separates those 2 chromosomes, this one here and that one here, separates those, and now you have 2 different cells. In one cell you have 1 chromosome, and the other cell you have another chromosome. After the next step of meiosis, meiosis 2, then you are taking this cell right here and you're separating it into 2 other cells. And you see they only have 1 copy of each chromosome. And here as well, it's separated into 1 copy of each chromosome. So you start with 1. After the first step, you go and you have 2. After the third step, meaning this third process, the third step of meiosis, the second step of meiosis, you have 4 haploid cells down here. You start off with a diploid cell, 2N, which is 46, and then you split and go to a N, a haploid cell, and then these are all haploid cells as well. Alright, let's move on. In meiosis 2, this is kind of just a diagram showing you more in detail. Remember, you had the 1 cell, which is right here, and it's split into 2. Okay? Because you separated these sister chromatids right here. And the big diagram is showing you up here. This is where the actual chromosome is, and it has 1 sister chromatid here and the other sister chromatid attached to it. Alright, so basically you're going to be separating those. And once you separate those, you'll have this thing over here, which is actually called a chromosome. You see this right here is a replicated chromosome, meaning it has its copy right next to it. And here is a unreplicated chromosome, where it's just the one chromosome, the one copy, without an attached copy. Okay, so these are the haploid cells you would have. All right, so in prophase 1 is just like it is in mitosis. You have pretty much a couple of things happening. We really focused on the chromatin coiling to form chromosomes as sister chromatids, so you can actually see them. And we also focused on the spindle fibers begin forming. So 3 and 4 is what you should really focus on. Meiosis 1 separates homologous chromosomes. Remember, we said meiosis goes through 2 steps, Meiosis 1 and Meiosis 2. So here is prophase 1. All right, prophase 1, just like in mitosis, you have in mitosis you had the chromosomes forming and the spindles forming. Right now you see the chromosomes sitting there, but these are homologous chromosomes, meaning they have this chromosome and then a copy right next to it. So we referenced tetrads before. A tetrad is after synapses happen, and synapses is the pairing and bonding of homologous chromosomes to form tetrads. So you take basically this chromosome right here and the one next to it, and those form together, and that forms a tetrad. Okay, so these things connected together and these connected together will give you 2 tetrads. And these homologous chromosomes that are, excuse me, trying to get the cursor up, right here, this one is from mom and this one is from dad. Same thing over here. This one's from mom, this one's from dad. And that's what we mean by maternal from your mother and paternal from your father. Homologous chromosomes. Homologous chromosomes consist of 1 maternal and 1 paternal chromosome. We just talked about that. 1 from mom, 1 from dad. They're the same size and shape and have the same genes on them. All right, genes. Genes are known to occur in different forms called alleles. We know this already. The eye color gene in humans, for example, occur in dark alleles and light alleles. 2 chromosomes can thus have the same genes but different alleles for those genes. All right, prophase 1. Crossing over. That's something that we haven't talked about yet because it doesn't happen in mitosis. Crossing over is pretty much the exchange of chromatid segments. You know, you talked about those chromosomes that have if this was just one chromosome by itself, it has one sister chromatid right here and then another sister chromatid right here. So once they cross over like this, they actually share and separate little segments, little pieces of those chromosomes, and they mix up. Okay, and then they end up looking. Actually, no, I didn't show it in that diagram, but they exchange chromatid segments within tetrads. So between these two, this one tetrad, they exchange chromatin segments to produce new allele combinations, and that gives you a lot more variety in the possibility of different traits that you receive. So the whole purpose is the crossing over end result. It increases genetic variability, the variety of genes. Okay, it gives you more variation or more diverse genes from crossing over. And crossing over is when those 2 sister chromatids cross and switch little pieces of segments. So they kind of separate and then rejoin again, but with different, with the other sister chromatid next to it. That shows the picture of chromosomes crossing over, and see how there's different segments now. Instead of them just being pink or blue, one's pink and has parts of pink or parts of blue in it. They're crossed over, so they separated and rejoined with the other. This is another diagram to help you understand crossing over. If this one chromosome on the left over here has A, B, C, D, all in capital, E, F, G, H, and on this other side they have capital A, B, C, D, E, F, G, H, the same. and then these two next to each other. One has lowercase all over them, one has capital all over them. Once they combine, and cross over, then you have this result over here. You have the one with the lowercase letters now has two. It has like a segment of the capital letters that came from over here. And then over here, the capital, chromosome has the two of the lowercase letters. So these cross over. All right, moving on to metaphase 1. Okay, metaphase 1 is pretty much just like it is in mitosis, except for you have the tetrads that are moving to the middle of the cell. These right here are tetrads moving to the middle, and then the spindle fibers are going to attach to the centromeres, just like they did in mitosis. Okay, actually, the term is a little different. The centromeres, like that right here, is a centromere, and that's a centromere. But when they are tetrads, they're connected at the kinetochore. That's what this word right here, the kinetochore of each centromere. So basically, that's where they attach. The spindle fibers attach there, still in the middle. All right, and then moving on to anaphase 1, tetrads. Basically the whole point is that in meiosis 1, we're dealing with tetrads instead of instead of chromosomes. Okay, so the tetrad is separated, and now you have one homologous chromosome going this way and one homologous chromosome going this way. That's anaphase 1. In telophase 1 or telophase 1, you pretty much have, the opposite of prophase happening again, where the chromosomes are starting to condense into chromatin, and the spindle fibers are forming. It says cytokinesis may occur, and two haploid cells containing sister chromatids are produced. So if that splits right there in the middle, then you have two separate cells. Okay, meiosis II separates sister chromatids. Meiosis II separates sister chromatids. Now meiosis I separated those tetrads, those homologous chromosomes. Okay, so here we go. Now we're on to prophase 2. Prophase 2, similar things happen. Spindle fibers form, chromatin coils to form chromosomes. What does this remind you of? Prophase 2 is basically the same as mitosis and prophase 2. I'm sorry. Prophase 2 is basically the same as mitosis, prophase times 2. So here you are, meiosis 2, we're talking about prophase 2. It looks the same. Now you have two cells, because remember the cytoplasm split right after telophase 1, and cytokinesis happens. So now you have two cells, one cell here, and you have homologous chromosomes in each. Okay, so now we're going to split these right here apart and just separate the sister chromatids. Metaphase 2, same thing. They all line up in the middle. Again, it's the same as mitosis, only now there's two cells doing it. Okay, so metaphase, they line up in the middle, they separate, just like this. They line up in the middle, the spindle fibers attach just like they always do, and they separate. In anaphase 2, during this phase, the centromeres of the chromosome and the two cells divide, and sister chromatids move to opposite poles of the cell. Long story short, very mitosis-like. Again, it's basically saying that it's the same thing as mitosis. Now you just have two cells doing it. They're separating. Now you no longer have those homologous chromosomes, but you have these unreplicated chromosomes over here. Basically, you just separated the sister chromatids. All right, finally, in telophase 2, you have the final act of meiosis. Chromosomes uncoil and back into chromatin, spindle fibers disappear, and then cytokinesis happens, and then you produce a total of 4 haploid cells. There you go. After telophase happens, you get cytokinesis that's going to come through and split this cytoplasm, split that cytoplasm, and then you have 4 new cells that are all haploid with just one copy of the unreplicated chromosome. Just like this, 4 cells, bow, 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 and there you are.